Have you ever noticed that two polymers with the same chemistry behave very differently in real life? One processes smoothly, another fails during scale-up, shows poor strength, or gives inconsistent drug release. Most of the time, the answer isn't chemistry, it's molecular weight distribution. That's exactly what GPC, or Gel Permeation Chromatography, helps us understand. In the next few minutes, I'll walk you through MN, MW, PDI, and retention time using practical lab logic, not textbook theory. GPC separates polymer molecules purely by size. A polymer solution flows through a column filled with porous beads. Large molecules can't enter the pores, so they move faster. Small molecules enter the pores and get delayed. As a result, molecules are separated based on size, not chemistry, giving us a full picture of polymer chain distribution. MN, or number average molecular weight, tells us the average size of polymer chains when each molecule is counted equally. MN is especially useful for detecting polymer degradation, monitoring batch-to-batch consistency, checking whether a polymerization reaction reached its target. If chains start breaking, MN drops first, which makes it a very sensitive quality indicator. MW, or weight average molecular weight, gives more importance to larger chains. Why does that matter? Because large molecules control material performance. MW correlates strongly with mechanical strength, heat resistance, long-term durability. For applications like medical implants, coatings, or high-performance polymers, MW is often the most meaningful number. PDI, calculated as MW divided by MN, tells us how uniform the polymer is. PDI close to 1 means uniform chain sizes. Higher PDI means a broader distribution. In real applications, lower PDI usually means better process control, more predictable performance. Higher confidence in regulated products like drug delivery systems and medical devices. Retention time is the time a molecule takes to pass through the GPC column. Large molecules elute earlier. Small molecules elute later. Modern GPC systems use advanced detectors like MALS and refractive index, improving accuracy and supporting regulatory expectations. Retention time trends also help identify issues like aggregation or degradation. In practice, GPC analysis follows 5 key steps. First, the polymer is carefully dissolved in a compatible solvent. Incomplete dissolution is a common source of error. Second, the solution is filtered to remove particulates that can damage columns. Third, the sample is injected into the GPC system. Fourth, separation occurs inside the column based on size. Finally, detectors generate data for MN, MW, PDI, and retention time, which are interpreted together, never in isolation. This holistic interpretation is where experience really matters. Let's look at where this analysis makes a real difference. In drug delivery, GPC tracks polymer breakdown inside the body, ensuring safety and controlled release. In polymer manufacturing, it ensures every batch meets design specifications. In biopolymer research, it helps understand proteins, sugars, and complex biological structures. And in regulatory filings, GPC data supports IND, NDA, and device submissions with defensible molecular characterization. This is where laboratories like ResolveMass come in. Accurate GPC analysis isn't just about instruments, it's about choosing the right columns, using appropriate calibration strategies, interpreting distributions correctly, explaining data in regulatory-ready language. From early research to commercial production, the goal is the same: clear, reliable molecular weight data you can trust. To summarize, MN tells you about chain count and degradation. MW predicts performance. PDI shows consistency. Retention time connects everything. Together, they give you confidence in your polymer, from early research to final product. Thanks for watching, and if you need expert support with GPC analysis, Resolve Mass Laboratories is here to help.